All right. Hello and welcome to day 2 of our workshop. Again, let me know if anyone has any trouble hearing me in this room or the other room. So just to give a brief overview of what we'll be talking about today, we have a lot of really cool, interesting topics. There's a bit of a variety today. We got to hold it closer this time? Okay. Um, and it'll be primarily focused in the beginning of the day on what do you do once you have called your variants. So we'll talk about the genome aggregation database first. Then we'll do a hands-on exercise that's reproducing a study in Terra. Then we'll talk about genotype filtering, refinement, call set evaluation. And at the end of the day, we'll talk about Dragon GATK and then about long-read sequencing. Okay. Um, and before I get started, I also realized that I didn't introduce myself yesterday. So hi, my name is Emma, and I have been working full-time at the Broad Institute for about 3.5 years. And I primarily focus on structural variants. So I'll be talking to you about those tomorrow and Thursday. But I also interned at the Broad Institute before I started working full-time, and I worked on gnomAD, and I continue to work on the structural variant side of gnomAD and the All of Us research program. Okay, so with that, I will get started talking about the genome aggregation database, and we abbreviate that as gnomAD to make it a little easier to say. So as Eric mentioned yesterday, from individual to individual, we share about 99.5 to 99.9% of our DNA sequence. So most of our DNA sequence is shared among individuals. But out of that 0.5 or 0.1% that differs between individuals, some of that is rare variation. Maybe only one individual in the world has that genetic variant, but the majority is common variation. Either it's common across the world or it might be common within a specific population. So these figures are showing some of the patterns of genetic variation across diverse global populations. And this is from the 1000 Genomes Project, which was one of the first efforts to sequence a large, diverse group of humans to start studying patterns of variation in the population. So the key reason that we're interested in creating these large databases of variation is that if you look at a single genetic variant in a single individual, it's very hard to say, is it part of that natural background population variation that's common in everyone, or has it even been seen in anyone else in the world? And is this variant potentially disease causing? It's hard to know that if you are just looking at a single variant in isolation. But we can start to answer those questions if we look at the genomes of many humans at once. So over the years, there have been many efforts to aggregate large data sets of human genomes, starting, well, this timeline starts in 2010 with the 1000 Genomes Project, and then it shows a selection of some of the projects that have gone on up until now. So including the Exome Aggregation Consortium, which was a precursor to gnomAD, and then several versions of gnomAD over the years as they've added more samples. And then towards the end is the All of Us research program, which is a US-based biobank funded by our National Institutes of Health. And we can see that over the years, these sample sizes have really drastically increased. The 1,000 Genomes Project was really huge at the time, but it only had a few hundred samples. And over time, these studies have aggregated larger and larger numbers of first exomes and more recently larger numbers of genomes as well. And they've also started to add different variant types, including copy number variants and structural variants. So earlier this year, both the All of Us research program and gnomAD released new versions of their call sets. And All of Us is now at almost 250,000 genomes with structural variant calls on about 10,000 of those. And gnomAD, well, we'll get to that here. gnomAD v4.0 was released actually earlier this month on November 1st, and now has over 76,000 whole genomes and over 730,000 whole exomes with both small variants, so SNPs and indels, and structural variant calls on those. So it's really big. It turns out it is the largest freely accessible public population genetics database out there. And importantly, all of these samples were joint called together. And none of these samples are known to have a Mendelian disease or disorder. So we expect that it is a pretty good representation of the normal population variation that is not causing severe Mendelian diseases. It's also relatively diverse in terms of genetic ancestry. So this figure is from gnomAD v3, showing the distribution of different genetic ancestry groups that are included in that data set. And the diversity has actually continued to increase with the addition of more samples in gnomAD v4. And you can browse this data set on a web browser at gnomad.broadinstitute.org. Just anyone in the world can open that link and look through this data. Okay, so what can we do with all of this data? These are some of the really important insights that we can gain. First is we can estimate allele frequency, both on a global level and in a specific population. So if you are a clinician trying to find your patient's cause of disease and they have a rare disease, you're probably only interested in looking at variants that are also rare. If they're common in the population, it's very unlikely that that variant could be the cause of your patient's rare Mendelian disease. So you can use the allele frequencies from gnomAD to filter your variants and remove variants that are too common to be the cause of disease. We can also look at variant co-occurrence, so which variants occur together in the same individuals and are shared on the same haplotype. So one downstream application of this is imputation. And we can also look at constraint. So where do we see that variation is depleted compared to what we would expect based on what we know about mutation rates? Where is there less variation observed than expected? And that, if we do find that there's a depletion of variation, that indicates that that gene or region is probably subject to evolutionary constraint. So if there is a mutation that occurs, it is probably not propagated within the population. It doesn't have a chance to become common because it's quite deleterious. So in this table, you can see it broken down by mutational consequence. So we have a line for synonymous, which is really just a baseline. We don't expect synonymous variation to really be constrained. And then we have missense and probable loss of function variants. And the gnomAD browser includes all sorts of information, both from gnomAD itself and from outside resources. And it's kind of all aggregated in one place, so you can kind of get a full picture of what's going on in the genome at that region. So some of the things that you'll see include variants that have been reported to be associated with disease in the ClinVar database. So in this top figure, we see a gene NSD1, and then each of the little red X's or triangles indicate a variant that was put into the ClinVar database and reported to be associated with disease in that gene. So you can see the exact position where all of those variants that are reported to be pathogenic fall in this gene. Next, there's regional missense constraint. So we were just talking about constraint. Often we talk about that on a gene level. So in this gene, is there less variation than we expect to see? But we can also look at that on a regional level. So is there a part of this gene that has less variation than we would expect? Sometimes there's just one domain of the protein that's really constrained against variation, and the rest of the protein tolerates some variation. So we can learn that with this metric. And the last thing on this slide is per base expression. So this is from the Genotype Tissue Expression Consortium, or GTEx. And it's saying in RNA transcripts that were sequenced from various tissues, how often is this particular base of DNA actually expressed in RNA in those tissues? So we can see that most of the bases in this particular gene are expressed all of the time, but there's a little bit at the beginning where those bases don't tend to be included in the RNA transcripts that were sequenced. And really the core functionality here is that with a large sample size, gnomAD enables us to estimate allele frequency pretty well. As we add more and more samples, we can get a more accurate estimate of how often this variation occurs in the population. And importantly, as the diversity of those samples increases, we can also we can get both a better estimate of a globally representative allele frequency and better estimates of allele frequencies within each population that's included in the database. And importantly, again, I'll highlight that all of these samples were joint called together, which enables us to have a genotype for every sample at every site and get these accurate allele frequency estimates. Okay, so I've kind of highlighted the purpose of gnomAD and some of what we can do with it, but now let's take a look at how it actually looks in the gnomAD browser. And I invite all of you to explore the browser after this. But for now, I'm going to just point to the slides so that I can clearly point out what I'm looking for and do some annotating on the slides. But if you go through this later on your own, each of the slides has a link in the bottom right corner to the exact page that we're looking at, so you can follow along later by yourself. Okay, so in this page of the browser, we're looking at the gene NSD1. There's some gene information on the top left, including, the ensemble transcript ID. On the top right, we're looking at that table showing information about constraint on a gene level that we talked about before. Along the bottom, we see the coverage in the exomes that are included in gnomAD in blue, and the genomes that are included in gnomAD in green. So we can see that the exomes only offer coverage across the exons. That makes sense. That's what they're supposed to do. So there's a lot more variation in the blue part of that plot. But the genomes have lower coverage overall, but it's far more consistent. They're also covering the introns. So that's what we would expect. But that information can be useful if you see that, say, there's no variation in a particular region. You can tell whether that means that there actually are no variants in the samples that are included in gnomAD in that region, or maybe it just wasn't covered by the sequencing for some reason. Way down at the bottom, there are these little toggles that say show transcripts and show tissues. So if we click on those, then we get this expanded view where we can see all of the known annotated transcripts that have been observed for NSD1 on top. And then we can see our per base expression levels, but broken down by each tissue. So if you are a clinician investigating your patient's cause of disease, you can zero in on the tissue that's relevant for that disease and look at the per base expression. Maybe the variant that you're looking at actually falls in a region of the gene that is not really expressed in your tissue of interest, and you maybe could rule out that variant as the cause of disease. There's another little button that says show transcript tissue expression. So we can see yet another view of this with transcripts on the y-axis and tissues on the x-axis. So this is telling us in each tissue which transcript was the most highly expressed. So that's another way to look at this. You can find out which transcript is really relevant to look at for your tissue of interest. Okay. Below that, we are looking again at variants that were reported in the ClinVar database. And we can see that some of those were reported to be pathogenic or likely pathogenic. Some of those were variants of uncertain significance, or there were conflicting reports of pathogenicity. And some of those were reported as benign or likely benign. They were ruled out as being causes of disease. And we can see how they're distributed across this gene. And we can also toggle which ones we're interested in seeing. So if we click on this button that allows us to only see pathogenic or likely pathogenic variants from ClinVar, then we get to filter out and only see the variants in red that are pathogenic or likely pathogenic. Next, we can look at what variants were actually observed in this gene in the samples that are part of gnomAD. So across the middle of the screen, we can see color-coded variants by their mutational consequence. So red for probable loss of function, yellow for missense, green for synonymous. And we can see how those variants that were observed in gnomAD are distributed across this gene. And you can see that actually there are variants all the way across this gene because we have so many samples in gnomAD that we're able to see quite a bit of variation. And below that, we can see the individual variants in a tabular format. So we can see the position where the variant occurs, what type of variant it is, what is the consequence on the protein, what is the variant effect prediction. So it's intronic, or it causes a frame shift, or it's a missense variant, that type of thing. And then we can see information about the allele frequency and a whole bunch of other stuff. Oh, okay. And then If we're only interested in variants of a certain mutational consequence, so here, let's say we're only interested in variants that we think are likely to cause loss of function of the protein encoded by NSD1, we can click this peel off only button, and we can filter to only variants that are annotated as probable loss of function. So we now only see red dots across the gene, and the table also only shows variants that are probable loss of function variants. And now let's take a closer look at one variant and see what we can learn about that one variant in gnomAD. So if we click on the first variant in the table, then we go to a whole page that's just dedicated to that variant. And we can see that it is an insertion of a single base, a C, at this position on chromosome 5. On top, we can see information about this variant's allele frequency across all of the samples in gnomAD. It's broken down by exomes and by genomes. So it's a little bit small, but you can see that the allele count is 1 in exomes. So it was actually only observed in a heterozygous state in a single sample in all of gnomAD. And at the bottom, we can see this information broken down by population and by sex. So we can now see that our one carrier is a South Asian male. We can see some more information about this variant. These slides are from an older version of gnomAD that was on HG19, so we can see information about this variant being lifted over to HG38. Then we have annotations from the variant effect predictor. So this is saying what is the likely consequence of this variant on the protein, and it's saying it performs that annotation for each of the transcripts for this gene. So in this case, for most of the transcripts for NSD1, this is a frame shift variant because we are we're inserting one base into the coding sequence. So all of the codons that are downstream are shifted by 1, and all of the amino acids after this insertion are kind of scrambled. They're different from what they were in the reference, and that will likely lead to early protein truncation as well. And so that also means that we annotate this as a probable loss of function variant. Down at the bottom, we see this LOF curation section. And actually, a lot of the high quality probable loss of function variants in gnomAD were manually curated to see if they are actually likely to be loss of function variants and understand that class of variation further. So you can see the results of that curation. It was curated as likely loss of function. Okay. Next, we can see some statistics about the carriers of this variant versus all of the other individuals in gnomAD. So you can start to look at, are there any differences between carriers and non-carriers that might be something interesting or that might indicate some sort of bias in the data? We want to kind of expose those statistics to users so that you can assess the variant quality yourself. So here we see the distribution of the age of the samples in gnomAD. So we see it's a kind of a nice normal distribution. And then this bar that's a deeper filled in blue indicates our one carrier. So we can see that they're around 50 to 55 years old. We can also see the distribution of genotype quality at this site for carriers and non-carriers. and actually almost everyone has a very high genotype quality, around 95 to 99, including our carrier. We can also look at site quality compared to other sites in gnomAD, and this site is pretty close to the average quality. We can even look at the actual reads that were used to call this variant in a selection of carriers. So in our one carrier for this variant, we can clearly see the insertion being called in a heterozygous state in this IGV that's embedded directly into the gnomAD browser. And importantly, this is using the haplotype caller BAM out. So it's the local assembly that haplotype caller was looking at when it made this variant call. Okay, so now that you've seen the gnomAD browser, what can we actually use this for? gnomAD is primarily used for variant interpretation and filtering. So one use case is that you can manually browse gnomAD through the browser. You can look at a small number of variants or genes. And this is really useful in a clinical setting. If you are trying to find your patient's cause of disease, you can go and investigate whether that variant was seen before, how common is it, what does the rest of the variation in that gene look like, all things like that. gnomAD can also be used to train variant filtering models, although that will drastically increase your runtime and compute resources, but it allows you to take advantage of the large sample size of gnomAD. And you can use gnomAD in functional annotation tools so that you can add metadata from gnomAD into your own call set. So for example, you can annotate the allele frequency of a variant in gnomAD in your own call set so you can compare them. Okay, so let's look at a bit of a case study. Imagine that you are a clinician and your patient has recurrent mouth sores, and their parents and grandparents and siblings also have these, so it looks to be something inherited genetically. And so you order whole exome sequencing, and whole exome sequencing gives you 67,000 candidate variants. So that's way too many. You're not going to be able to find the cause of disease for your patient from that many variants. So what do? You can start by using the gnomAD allele frequency to filter to rare variants. This is a rare condition, so you expect that it's caused by a rare genetic mutation. So you can remove all of the variants that are more common than 1% allele frequency in gnomAD. And that gets you down to just 7,000 variants. From there, you can filter based on the variant quality, and that gets you 4,000 higher confidence variants. And then you can look in ClinVar to see whether any of those 4,000 variants have already been reported as pathogenic or likely pathogenic for the condition that you're interested in. And it turns out that in this case, there are only 6 variants that were previously reported as pathogenic or likely pathogenic, and 6 is a much more manageable number. So you're in pretty good shape now. You should be able to find your patient's cause of disease. Okay, so just to bring this back to the GATK best practices variant calling pipeline that we were discussing yesterday, this part at the end is really where the gnomAD dataset fits into that. It can be used for variant filtering, refinement, and annotation. And that's all I have on gnomAD. Any questions? Yes.
I would like to know the function of the gene constraint board. because we only see someone use it to evaluate the de novo mutation. But it raises me another question is about will we use the gene constraints or to predict the inheritance pattern, inheritance autosomal dominant or chromosomal recessive or gene or penetrant?
Okay, let's see. So your question is about how you can use the constraint metrics.
Yeah.
So I'll start on a more general level. If a gene or region is constrained, then we expect that variation in that gene or region is more likely to be deleterious. So one use case is to kind of prioritize genes or regions as potential causes of disease if they are more highly constrained. Um, and I think it could be used for dominant inheritance modes as well as for de novo for sure. Does that answer your question?
Yeah.
Okay. Yes.
Mention the gnomAD variant as joint coding together. And I'm wondering, because we only have different sequence, different technology format or sequence of genome sequence, how you join coordinate together? What is the concern?
Yes. So thanks for asking that because I think I wasn't totally clear. Actually, the exomes are joint called with all of the other exomes, and the genomes are joint called with all of the other genomes. But the exome and genomes are not joint called together. Right. Right. So that's why the browser always contains independent information from exomes and genomes because those are actually different call sets.
Question is about the depth. Because all different sequences and different sequences formation have different depths. Each different sequence. How concern.
Yeah, so the question was what okay, next time. Yeah, next time I can just bring the mic. So the question was what is the how, so there's variation in sequencing depth from sample to sample, and how does that impact the joint calling? Um, and so I guess the short answer is we're still able to do high quality joint calling. I'm not sure if Eric or anyone else wants to add to that. No? Okay. All right. Yes. Maybe I'll just bring the microphone to you this time.
I'm just curious about gnomAD, or if I pronounced it wrong or not. This database is how to control the quality of those variant we are going to include in this database.
Okay. So, what kind of quality control was done for gnomAD?
Yeah,
Or
yeah.
okay. Yeah, I mean, it was very heavily filtered and evaluated because we wanted to produce a high quality data set and make sure that the precision in particular is really high, so we're not publishing a bunch of false variants. So throughout today, we'll be talking more about some of the filtering methods that are used in GATK, and that might answer some of your question about that.
So the course today about filtering by using GATK might maybe this method are used in this database.
Right, yeah. So we'll learn more about variant filtering. Right. The next session will be a hands-on exercise, but after that, we'll be talking about variant filtering as well as call set evaluation, and that is all relevant to how gnomAD was created.
Thank you.
Yeah, thanks. Oh, okay.
One more question. Sometimes we can see here the filter is not passed. How do we use the information?
Okay, yeah. So when the filter status in the browser is not pass, that indicates that, that variant was not super high quality. So we still want to publish those variants that weren't that didn't have a pass filter status, because those might be artifacts, and you might actually see those artifacts in other data sets.
Yeah.
And that knowing that that was a variant that didn't pass filters in gnomAD could tell you that you want to be a little bit more careful about that variant in your own data. Alternatively, if you are a clinician who is looking into that variant as a potential cause of disease, you might go to gnomAD, and you don't want to see that that variant is completely missing, because then you would conclude that's a really rare variant. Instead, you might see that it's present, but it didn't pass filters, and then you might draw a different conclusion. Maybe it's an artifact, or maybe it's a variant that does occur in the population, but just wasn't super high quality in gnomAD. Yes. Okay. Lots of questions.
Thank you. So I want to ask for rare alleles that they have very, very, very low frequency in the population. How do you usually define a cutoff to like a reliable cutoff? Because I see sometimes it's 0.1% or sometimes it's 1%. And to be confident enough that's not a false positive. Yeah, that's my question. Thank you.
Yeah, good question. There was actually a method for setting a filtering cutoff for rare variants for rare diseases that was published. I forget the paper that was in. I can try to find that for you, but there is a published method for determining an appropriate allele frequency cutoff depending on how rare your disease is and the inheritance mode. Yeah. Yeah. Oh.
I'm just wondering, is the functional filter mainly focused on loss of function, but sometimes gain of function may cause disease?
Yeah, so we definitely don't want to ignore variants that are not annotated as loss of function. Plenty of variants that are annotated as missense or something else could be disease causing. So a really good variant interpretation pipeline would take that into account and consider variants that are not loss of function.
So now it's only loss of function.
I think clinicians generally do consider other mutational consequences depending on what's known about the disease. So some diseases are specifically caused by loss of function variants and some are not. So a clinician would take that into account when deciding which variants to look at. I was really just illustrating in the browser that you can choose which variants you are most interested in, but you could certainly also select missense variants for that. All right. I don't want to put us behind time much longer, so I think we'll pass this off to Anton.